First of all, I want to thank the organizers for this meeting, and to put it here in Nijmegen, which was my first city when I started my postdoc, and I took the bike here, and great memories to be here again. So, and the second thing that I want to say, I want to ask the indulgence of the primate researchers here, because this project has nothing to do with, well, it has to do with human primates, but not with non-human primates, and but it's here in any case. So I will present the project and the results. So, as the project states, we wanted to study development at the micro scale and the micro scale. That means at the level of the cortical microcircuits, and also at a more phenomenological level, in order to establish then what happened when the subjects are confronted to multisensory stimulation, and how then the system, which is in these development stages, can start processing stimuli coming from different domains. Our project is a collaborative instance between the University of Amsterdam, in which I am, together with my colleague Umberto Cese, and my colleague Luca Monteliciani, who is here, the Vision Lab in Pisa, Pantheon University in Greece, and the Center for Brain Research in Lyon, I think, France. So, I wanted to say also that I've been working with the Partnering Project environment since almost their inception. In 2016, also with my colleague Umberto Cese, we won a project called Canon, in which we studied in a more fundamental way the integration at the level of a cortical microcircuits, and we try to do that also by using some sort of interspecies comparison, going from mice to ferrets, and to use also data from monkeys and humans. And that project, which I'm not going to present here, but I'm happy to discuss with you in other instances, set the necessity to go into the development and to see what happened at these early levels in when you present those stimuli from different senses, in order to for the brain to start establishing a sense of reality. And also we wanted to make it in a more applied way, to understand why, if there's any multisensory impairment in this development stage, that could have a potential role in the perceptual and cognitive impairments that you can see in patients with neurodevelopmental disease, such as like the autism spectrum disorders. And in this cartoon from Baum and Wallace in 2005, they already showed that these sensorial and multisensory representations are very important to establish the basis of the perceptual and cognitive abilities of normal individuals, and of course that impaired ability to properly integrate these multiple modalities can underline to be the cause of many symptoms in autism spectrum disorders. Yes, and that also can affect other type of symptoms, like in language, and also communication, and the capacity for social interactions. So the thing is, we don't exactly know how the multisensory integration capability develops in the early brain, and how that is expressed in the autism spectrum disorders. So, and just to give you an example, so here you see another paper from Phosphate in 2010. It's a behavioral result in which they show that subjects, patients with ASD, compare with the normally developed or typically developed kids under the simulation of a two-flash illusion paradigm, so a paradigm in which you present two flashes of light together with a sound. Well, and in some cases you presented one flash and the sound, then you get the illusion of that there is two flashes at the same time. So by using this paradigm, they found that the way that they modulate the stimulus onset, the synchrony between these two modalities, they found that the autistic spectrum disorder has this sort of increased window of integration between the synchronicity of the two sensory stimuli. Compare that with the normal subjects. So then the question is how that can occur and how we can understand and provide some neuronal correlates and some neurophysiological basis of that phenomenon. So the domino paradigm, as we did call it, it uses three, let's say, it's focusing on three levels. So one level is these experiments in mouse, in which we try to do a multi-area characterization at the level of the microcircuits. That means we try to go into the cortex and other areas, isolate spiking activity and local field potentials, and characterize the development of wild-type mice and a model of autistic spectrum disorder. And I try to do that from early onset until until the adulthood. That combined with human experiments in mostly psychophysical experiment, but also including EEG in subjects with ASD, but also normal subjects, and also try to do these experiments on also, manipulations of multisensory integration from childhood to adulthood. And finally, and then it's where EBRAINS can be relevant for us, is to try to use this data as an entrance point to modeling experiments in which we try to use spiking neural networks with a biologically plausible plasticity and use those models as biologically plausible as possible as a model of multisensory integration development. So how we interact to each other? Well, we have these three work packages here, and of course you see arrows going to everywhere in the way they're trying to organize and coordinate the experiments in such a way that that can be as much compatible as possible, providing all the limitations that you can have by having two different models. And of course then go into the modeling steps. But of course we would like to then interact with EBRAINS and the human brain, and once we have that models and that integration of the data, go into EBRAINS and use the EBRAINS infrastructure for that. And I will refer to that at the end of this presentation. So I'm going to give you a very brief overview, very not focused on details of what we have so far. The project started in 2020. We have still, and it will finish at the end of 2023, so we still have time to go further, and that's when I'm going to refer now. The team, as you can see here, so I'm working with Umberto Cese and Luca Monteliciani, and here you have Argiro Vatakis and Pantone University, Guido Marco Ciccini from Pisa, and Vinod Cotterell from in Lyon. We are focused on mouse experiments, and I would say we as University of Amsterdam here. And what we're trying to do is to have a model of mouse model in which we can record them while they're passively viewing and listening stimuli that are presented on the screen, and we record them in different brain areas using neuropixels. And neuropixel is a nice technological advancement that allows you to have these very high density electrode recordings with a very high signal-to-noise ratio that give you not only the LFP signals, but also the possibility to obtain a large number of neurons. And by having this combined with the histology and the precise localization, we can have a good estimation of the functioning of that multiple areas. Now, what we are doing here is an experiment that, and I agree with you if you are thinking on the limitations that could have, to mimic in certain way the double flash illusion in these animals. These experiments, I would say, are passive, and what are focused are in the way that this animal passively can process the information coming from full contrast gratings presented on a screen, and a tone that is presented with a particular stimulus onset asynchrony from the visual stimuli. And what we want to do this, we wanted to do it in wild-type mouse, but also in mouse with genetically modified, in order to have with Krylov systems to optogenetically control the particular interneuronal populations, parvalbumin, VIP, and SST. And why we can do that? Because one of the theories that we wanted to prove in this whole idea of the neural development of multisensory integration is how this excitatory and inhibitory relationship between the components of the cortical microcircuits are interacting in these animals. And then we are using a knockout mouse from the protein F, which is a model of Fragile X syndrome that has been also equated to in clinical setups as patients with autism spectrum disorders. The Fragile X patients has resembled several characteristics of the ASD, and also they have that problems on multisensory integration and socialization and among other cognitive disabilities. So we use these combinations of animals, and one of the ideas is to combine, we are not yet there, but to interbreed these different genetically modified lines, so we can have ephemeral knockout mice with also a Krylov control that we can use to optogenetically control the population of interneurons that are presented here during the passive presentation of this stimuli. And we want to do that in early development, starting from P15 and taking in consideration several neurodevelopmental milestones on the development of the cortex of the mice. And if time allows us to do it, as so far as been, thanks to Luca, the wonderful postdoc that is in charge of this experiment, to do it also during in normal light conditions, but also to have the animals in dark rearing conditions, which is a typical way to manipulate the neurodevelopment, early neurodevelopment. So far, what we are doing, we have having these recordings, and we have been able to have this firing rate of the cells after the presentation of stimulus. And here we have some examples of what happened when, for instance, there are presentation to a flash. You have the increase of the spiking rate, but also you see that, for instance, the presentation of the when you have only sound, or when you have the sound flash overlap as a function of the stimulus onset asynchrony. So in black you have the visual response, which is kind of what you have to see here, and then here you see the different stimulus onset asynchrony, and you can see how then the super additive effect when you have a very short stimulus onset asynchrony between these two stimuli. Now, then the next, while we're setting those experiments, then it comes the question, how do the temporal aspect then vary across development and in during normal development and in autistic spectrum disorder? So we are going to use, as I say, this ephemeral, this
Can I ask a question?
Yes, please.
Do I interpret that correctly, that your response to sound in V1 is equally high as to visual stimulus?
I think this, yes, but this is not an average of the whole population. This is one example of the highly responsive neuron. Yeah, and we use that to compare the other responses. What I wanted to say that we have this knockout mouse model of Fragile X, and why we choose that, it's also because in a previous work that I did together with Tara Arbav and Francesco Battaglia, who is here at the Donders and a part of the previous Canon project, we showed that in hippocampus that there is this abnormal hypersynchrony when the animals are just freely walking in a round maze. And very briefly, you can see here these signatures of hypersynchrony. One of those is this increase of the power compared with the wild type in lower frequency, in the theta frequency in the hippocampus, and increase of the noise correlations across multiple windows of integrations during this process. And also, you see here a comparison between pyramidal cells and interneurons of the spike field connectivity between and the comparison between the mice and Fragile X and normals. And you see that these animals are more, so the neuronal assemblies are more synchronized and cells are more locked into their rhythm that normal the counterparts, and that might reflect functional deficit in local networks. And that is something that we want to explore also in the visual cortex in these other mice. How much time we have? 5 minutes. Okay, then I'm going to go very quickly to show other results which are very important, which are related to this core paradigm that I mentioned before. One of those is made by my colleague Umberto Cese, together with another student, Matthijs Lood van Huis, which shows in head-fixed mouse also this is a normal wild-type mouse without any other not knockout mice. Also, a multisensory integration. In this multisensory integration paradigm, here is active, and the animals need to do a decision based on whether the sound is incongruent or not congruent. And when animals were trained, then the idea was to focus higher in the hierarchy, so to go to the PPC, an area that, of course, has been it has to do with multisensory integration, but also decision and action. And to make long story short, they have this audiovisual change attention task. The animal needs to detect that change, and then what they were trying to do was to see whether if there's any effect, if while using optogenetics, they use inactivation either on V1 areas or higher in the hierarchy like PPC. And while they found that you can have an inactivation and a lack of multisensory integration in the visual areas, when you go to PPC, you will see that normally you have in these areas a correlates of what they call everything. The animals can correlate, or there are correlates based on these models here, just to show how is the response and isolate the response across the multiple type of stimuli, that they respond to visual activity, they respond to the congruent or the incongruent, they respond even better when they are well trained compared to naive, and they also respond when they are a correct decision or not. So they conclude that the PPC reflects these sensory informations and predicts task performance. But the interesting thing is that there is no causal role. When there is a bilateral PPC inactivation with optogenetics, what they found is that, and after trying to correct by Bayesian t-test, trying to really show the absence of an effect, they were able to conclude that no matter the inactivation, the still animals they can follow that behavior, no matter so you see here in D prime, when they are detecting the two modes of stimulation, the reaction time, and also other parameters. So the conclusion of this task was that we need to be cautious in the way that how animals or how parietal cortex is involved in multisensory integration. It's not a one-to-one way to interact with behavior and with lower visual areas, but if they are involved in the processing, but it requires more to the PPC, probably other areas like prefrontal cortex, to have a causal role in the integration of multiple modalities. Our current activities in the project is continue the recordings of specific neurodevelopmental milestones, use the comparison with dark reading, and do the recording with combined genotypes. And of course, at some point, we will focus on data analysis and go to my expertise in LFP, spike data, correlations, and comparison. In the 1 minute that I have, I'm going to briefly mention the other two work packages, because they are working with humans, and the way they are doing is in dual ways. One is to study normal subjects in these psychophysical tasks that try to show how, in this case, in unisensory approach, how they can report the difference of a particular target stimulations when they are crowded or it's full of distractors. And they are doing this by having a low reliability target, so a target that is very ambiguous, or a high reliability target, so a target that you really can differentiate from the crowd. And here, what they study is to show how this, the response of the flankers orientations and the bias of the response. And what it's the take-home message here is that this integration of the information from the centers and the periphery occurs early. There is no necessity to go hide in response, or they claim that occurs everywhere, and it's also very much adapted to these difficult presentations instead of others. And just to finish, and just to mention, the second part of the work package 2 is to develop tasks that can be used in autistic patients in order to improve the diagnosis based on this multisensory integration task. They are using the McGurk effect in a task in Greek in order to see whether the patients can detect this sort of combination of syllables when they are presented on, and then we need to read it from the face of the lips of the of the counterpart. And what they found then is that they have difference between the autistic spectrum disorders and the normals, and they can see that systematically these patients can fail to detect this illusion. They are still doing this, and they want to go more into the linguistics aspect and see how that there are specific questions about place and manner of articulation of the McGurk illusion, and how that can be used not only for the specific Greek and cultural aspects of the Greek language, but in a more universal way. VP3 or computational model, I'm only going to mention that is under development. We have already a model, spiking model of the visual input. There is a development of a model of the auditory input, and it's part of the project to do it, to go to the audiovisual model in the last stage of this process. Our future developments, of course, using EBRAINS, needs to go within the data sharing and curation, and one of my aspects to be here is to learn about to do that, and also to implement this computational into models into the large-scale models, and also where we can use the whole tools to improve our data integration between mouse and humans. And with that saying, I'm sorry for the delay. Thank you very much.